So, hello everyone. So, welcome to the first module of this online course Fundamentals of Bioprocess Engineering. So, in today's lecture we will discuss overview of fermentation process. So, fermentation we are aware this is traditionally derived from alcohol fermentation. So, it used to be considered as an anaerobic process, but later on we will see in this lecture this fermentation can be aerobic, anaerobic, even it can be facultative. So, we will be seeing fermentation and different type of fermentation, then what is bioprocess and what is the role of bioreactor in the fermentation process. Further, I will briefly summarize the history of bioprocess engineering to just see the significant development in this domain. Then finally, I will list a few bioprocess products of commercial importance. So, as we know, the bioprocessing is widely used in food, chemical and pharmaceutical industries, and as I say, this bioprocessing means we are utilizing the biological agent, it may be the microbes, animal or plant cell or enzymes to produce useful product. Apart from this industrial products, it is also exploited for the wastewater treatment. Wastewater treatment. Now, you can easily correlate the wastewater treatment can be anaerobic digestion or aerobic, that is activated sludge process. So, it indicates the fermentation is aerobic as well as anaerobic. So, as I told, this originated with fermentation technique in ancient food production, particularly in anaerobic condition there was synthesis of alcohol, but now this spans from industrial alcohol to organic solvent, antibiotics, therapeutic proteins and vaccines. So, from the low cost to the high value products, all of them are being produced using bioprocessing. So, traditionally used to call this fermentation as an metabolic process where organic substance, let us say sugar, breaks down into simpler compound generating ATP, and this simpler compound or let us say simple sugar is further utilized to produce gas, acid, alcohol and is widely used by microorganism in food and beverage production. So, the traditional concept was to produce ethanol from the sugar. So, that term was being referred as fermentation. So, let us say glucose is converted to pyruvate and you just see the word fermentation without oxygen. So, in the case of anaerobic fermentation, this ethanol is produced and in the presence of oxygen it goes to the Krebs cycle. So, the detail of the process was we considered the anaerobic process, and in the anaerobic process it also produces the energy output in terms of ATP, converts NADH to NAD using pyruvate from the glycolysis, like sugar is converted to ethanol and proteins are converted to peptides and amino acids. And different species has been exploited for fermentation process like bacteria, yeasts, molds like lactobacillus, Saccharomyces cerevisiae and penicillin. So, you can see here the glucose is converted to pyruvate, in term this 2 ATP is also generated, and 2 NADH is converted to 2 NAD plus, and further this pyruvate can be utilized for the production of different metabolites like lactic acid using lactobacillus, ethanol using Saccharomyces, acetic acid using Acetobacter, propionic acid using Propionibacterium, and butyric acid using Clostridium species. So, now we will quickly see the few common fermentation process. So, first one is lactic acid fermentation. This is an anaerobic fermentation process where glucose is converted to lactic acid, and some biological agent is Lactobacillus streptococcus. In fact, in the animal muscle cell under low oxygen condition, when we do excessive exercise, there is a synthesis of lactic acid which causes muscular pain. Alcohol fermentation, this is also called ethanol fermentation, again anaerobic process where glucose is converted to ethanol and carbon dioxide, and the producer microorganism is yeast Saccharomyces cerevisiae, and some bacteria also like Zymomonas mobilis. So, here glucose is converted to ethanol, carbon dioxide and ATP. So, 1 mole of glucose results in the production of 2 moles of ethanol. Butyric acid, similarly, this is again anaerobic process carried out Clostridium butyricum or Clostridium acetobutylicum, where glucose is converted to butyric acid, hydrogen, carbon dioxide and ATP. So, glucose is converted to butyric acid, carbon dioxide, hydrogen and ATP. Acetic acid, so this is an aerobic process where now ethanol is oxidized to acetic acid by using acetic acid producing bacteria such as Acetobacter or Gluconobacter species. This process is commonly used in vinegar production. So, ethanol is oxidized, here you can see aerobic process means oxygen is utilized and acetic acid is produced. Propionic acid fermentation, the producer organism is Propionibacterium and Clostridium propionicum. It utilizes sugar or lactate as substrate, and the sugar is metabolized by EMP pathway to form pyruvate, and further pyruvate is first converted to oxaloacetate, then malate, fumarate, succinate, then finally propionate. So, main product is propionate or propionic acid, acetic acid, carbon dioxide, glucose is converted to propionic acid, acetic acid, carbon dioxide and water. So, this is the main pathway for the production of propionic acid, where along with propionic acid, acetic acid is also synthesized, and pathway I have already discussed. Pyruvate is finally converted to the propionic acid. Another common fermentation is diacetyl and 2,3-butylene glycol fermentation, where it starts with the citric acid, it goes to pyruvic acid, then it forms acetyl methyl carbon, and it can either go to formation of diacetyl or it can go to formation of 2,3-butylene glycol, and the producer organisms are Enterobacter. The reaction that leads to the production of 2,3 butanediol involve a double carboxylation step. In addition, you can see this bioprocess can occur in the presence of oxygen as well. So, here oxygen is actively supplied and consumed as part of metabolic pathway, and aerobic fermentation is mostly used for biomass production like microbes, Saccharomyces cerevisiae yeast and E. coli, and typical products are single cell protein and food supplements. Antibiotics are again produced in aerobic condition. Producer microorganism is Streptomyces, example is penicillins and streptomycins. Enzymes are aerobic fermentation products produced by Aspergillus fungus or Bacillus bacteria species. For example, amylases, cellulases, proteases, and wastewater treatment, as I told, particularly for biological wastewater treatment. This utilizes the mixed aerobic consortia and it produces carbon dioxide, water and biomass sludge. Now, we are going to quickly see the history of bioprocess engineering. So, it is divided based on the timeline. It says pre 19th century, 19th century, early 20th century, mid 20th century, late 20th century and 21st century. So, earlier it was early fermentation practices. So, technology was natural fermentation using yeast and bacteria, and few products were breweries like beer, wine, bread making and cheese production. In 19th century, this is also known as birth of microbiology. So, here the technology were fermentation technology, and it was explored to understand the microbial role in the food and medicine. In early 20th century, the key development is industrial microbiology, and one example is large scale fermentation, particularly penicillin discovery during World War II. Application is antibiotic production, organic acid synthesis. Mid 20th century, that is known as biochemical engineering era. So, it led to the bioreactor development and enzyme technology, and application, if you see the mass production of drugs, vitamins and bio based chemicals. Late 20th century, this is the rule genetic engineering resolution. Many recombinant strains were produced which can produce any novel product, or it can enhance the yield of existing strain. So, it was used for the production of biopharmaceuticals like insulin, growth hormones and synthetic enzymes. And in 21st century, it is the advanced bioprocessing, it is CRISPR and AI driven biomanufacturing, continuous bioprocessing, and potential application include precision medicine, biofuels and tissue engineering, particularly in biomaterials. So, if you see this pre 19th century, the natural process, these early humans unknowingly relied on wild yeast and bacteria to ferment food and beverage like wine, beer and cheese. The fermentation occurs naturally through environmental microbes. For instance, grape juice turned out to wine and malt fermented into beer. So, here you can see Aspergillus producing lactic acid, Lactobacillus producing lactic acid, Saccharomyces producing ethanol and bread. Now, in 19th century, this Louis Pasteur, a microbiologist work, raised the new set of research questions such as where do the fermentation agents come from, where do they originate from, germs similar to themselves or they do appear spontaneously as explained by spontaneous generation theory. So, it says if you are having container, open container, gauze covered jar and sealed jar, and you are going to observe different observation after certain time intervals. Why it is happening? Because the presence of microbes which is coming from the ambient, and they are able to utilize this substrate. But if it is sealed, you will not see any flies, right. So what he did in one case, the broth was boiled for 1 hour to sterile and allow them to cool down in the fresh air. So all broths are sterilized, and this was open in the air, and it you can see it become turbid, means there is a bacterial growth. So what it says is the air contains microbes which can utilize the substrate and produce some metabolites. You can put the cotton plug, it is blocking the bacteria from air. So it is working like a air filter, and if you can, bacteria were removed from the air entering into the flask by setting up a long neck. So ultimately, if you can block the entry of microbe, there will be no growth. So it says whatever fermentation is done, that is due to some biological agent. This is interesting story of penicillin. As I told, it started during this World War II. So it was very interesting story. This Alexander Fleming, he discovered penicillin when he observed a mold that was identified as Penicillium notatum killed surrounding bacteria in a petri dish. So he was doing this. He was isolating Staphylococcus aureus from the sample, and it was observed that a hollow zone was there. So here there is no growth of bacteria. So as there was no growth, so he further analyzed this particular region and found that because of this Penicillium notatum, bacteria was unable to grow, and it was further discovered that it produced some chemical which is killing the bacteria, and that is penicillin. Further in 1940s, the Florey and Chen successfully isolated and purified the penicillin, and they demonstrated their ability to treat bacterial infection, but still the large scale production was not materialized. Further, the engineering innovation for mass production, as I told, during the World War II, to meet the wartime medical need, scientists partnered with chemical engineers to solve the production problem. The engineers developed the deep tank fermentation, which allowed penicillium molds to grow in controlled liquid environment, vastly increasing the yield. So you just see it started in 1928 during the isolation of Staphylococcus aureus. In 1941, Chen, Florey and Hartley conduct the first human trial of penicillin during the World War II because it was needed to treat the patient. In 1945, the penicillin was used to treat patient on a large scale. So initially the penicillin strain was Penicillium notatum, but further to enhance its yield, another strain was isolated, that is called Penicillium chrysogenum, which had better productivity as compared to Penicillium notatum. And in 1950s, penicillin becomes widely available and revolutionized the treatment of bacterial infection. So if you just see the yield of penicillin it was 0.001 gram per liter in 1939, and now it is over 50 gram per liter in the fermentation broth. So, this is the revolution of bioprocess engineering. You can produce any metabolites and you can significantly enhance its titer or yield. And these are the few prominent companies like Merck, Pfizer, Squibb, which are producing penicillins. Further, this is just a flow chart. You can see here, this is your seed fermentation. It is going to your bioreactor. So, here there is a production of your penicillin. So, up to here this term is called upstream till the bioreactor operation, and from here you have to separate the cells, then you have to purify your product. This whole process is called downstream. So, you can see this is a flow chart including this production as well as the separation of the cell, then extraction and purification of your final product. So, interestingly, during this discovery you just see 3 Nobel Prize were given in this journey. First was awarded in 1923. It was awarded jointly to Banting and John McLeod for the discovery of insulin. Further, this Frederick Sanger won the Nobel Prize in Chemistry in 1958 for discovering the fully sequence of bovine insulin, and the third one is this Hodgkin was awarded Nobel Prize in Chemistry in 1964 for discovering the physical structure of insulin. So, this insulin is another metabolite which is being produced using microbes, that is recombinant bacteria or the recombinant yeast, and the whole process you just see the journey how it started from 1923 to 1964. It resulted in the 3 Nobel Prizes, and now insulin is commonly used for the treatment of diabetes. After seeing those conventional fermentation, now we can define what is bioprocess. It says it is a metabolic process driven by microorganisms that converts your substrate to the product, and this biological component can be microbe, it can be the animal and plant cell or component of cells such as enzymes. So, one can be product and other can be destroy harmful waste or the biological waste treatment. Other example, one can say the bioremediation. So, what is bioprocess? One can simply say the application of biological science in industrial process is known as bioprocess, and for the bioprocess when we are growing cells on a large scale, that is called industrial fermentation. So, industrial fermentation means you are going to use a bioreactor to grow your cell to produce your desired metabolites or products. So, here you can see this is a stirred tank bioreactor, this is the lab scale bioreactor, and this is the pilot plant bioreactor. So, scale keep on increasing from the lab scale to the pilot scale to the industrial scale. And as we have discussed in the bioprocess, you are going to have upstream, then bioreactor, then downstream. The upstream means you need to have the producer microorganisms, you need to have media and media optimization, further media sterilization, and what is the your limiting substrate and what is the feedstock. In the bioreactor, you can have this microbial process or the enzymatic process. In the enzyme, you can have free enzyme or immobilized enzyme. Microbial process, you can have the whole cell or recombinant cells. and then after production of your metabolites we will go for downstream, which will have a cell disruption in the case of intracellular product. Then finally you have to go for solid liquid separation, then you have to concentrate your product, purify your product, then formulate, and in addition whatever waste is generated, that also need to be treated. So now you can understand the major bioprocess products in almost all the domains of application of our daily as well as the essential requirements. So you can list the areas of chemical, pharmaceuticals, energy, food and agriculture. Chemicals, I have given few examples like ethanol, acetone, butanol, organic acids, acetic acid, butyric acid, propionic acid, citric acids, enzymes and polymers. In pharmaceuticals, it is started with antibiotics, different enzymes, enzyme inhibitors. Now, these monoclonal antibodies are being produced, steroids, and we all see the importance of vaccine, which is being produced by through bioprocess. Energy, I have listed two, that is the ethanol and methane. Food, very common, cheese, yogurt, baker's yeast, beverage, wine and beer, food additives, even amino acids, vitamins and proteins. These are various application in the food sector. For similarly in the agriculture sector, animal feed, waste treatment, vaccines, microbial pesticides. This is very interesting, this eco-friendly control of pest, and mycorrhizal inoculants. So you can see this is the vast area of application of bioprocess, where the fermentation technology is applied to produce at the scale which is needed for its commercial applications. So with this I will stop this chapter. So today we discussed this origin of fermentation, then we saw a couple of example, antibiotics and insulin, and further then we redefine the bioprocess and listing important bioprocess products. Thank you so much.